To compare the three CERNA treatments, the normalized gene counts have been converted into log-2 ratios against the control group. Each group has 2 replicate samples. Open the filter tool in the basic plugin. We will exclude genes that have only unstable measurements in the low count region. The threshold is not set as a fixed value. Instead, we decided while looking at a scatter plot of the gene counts. Here, we will exclude genes that have gene counts below 20 in at least one sample in every group. Check the group of genes to be excluded, click the Filter Out button, and save the remaining genes as the QC1 list. Set the QC1 list in the filter tool, and narrow down the genes further from here. The next genes to exclude are those that show almost no expression change. This filtering is not essential for DEG analysis, but it is essential for clustering. Again, we decide the threshold while checking the data in the scatter plot. The magnitude of expression change differs greatly depending on the dataset, so this threshold cannot be determined without looking at the data. We exclude genes that never move outside the region that can be regarded as essentially unchanged. In this dataset, we will exclude genes whose log-2 ratio against the control group never goes outside the range of plus or minus 0.3. Now the data are ready for analysis. First, we use PCA to get an overview of the expression profiles. In the PCA plot, the 2 control samples are located near the origin, and the CERNA-treated samples generally move toward the lower right. Psi1 is closest to the control, while Psi2 and Psi3 move farther away. Next, we perform tree clustering and visualize the expression patterns as a heat map. As we saw in the PCA plot, Psi1 has weaker colors, indicating that it shows the smallest change from the control group. In contrast, Psi2 and Psi3 each have genes that increase or decrease strongly in characteristic ways. In this way, PCA and heat maps should not be interpreted separately, but in relation to each other. Next, we extract genes whose expression changes in each CERNA treatment compared with the control group. Thinking of the comparison as a fraction, drag and drop the reference group into the lower box. After setting an initial threshold, check whether it is appropriate using the visualization tool. If this threshold seems a little too conservative, try relaxing it. Once the threshold is decided, extract the DEG list and save the up and down lists separately. At this point, it is useful to organize them into folders so that you can tell they were extracted from the QC2 list. Check the extracted DEG list using the visualization tool. In the Compare Two Groups tool, keep the control group setting as it is, change the group to be compared, and extract DEG lists in the same way. Using the Venn diagram tool, which is a basic function of Subio Platform, create a union list of all DEG lists. With the hashtag overlaps tab, you can set 3 or more gene lists at the same time. By applying tree clustering to the union list of DEGs, you can check how similar or different the expression changes are among the treatment groups. By clicking each DEG list, you can visualize whether the same genes behave similarly in the other groups, or whether they show different patterns. The Venn diagram also shows that the up-regulated DEG lists have a relatively large number of common genes. In contrast, the down-regulated DEG lists have fewer common genes.